With a diameter of 59.1 inches, this London plane tree is the largest planted tree at the New York Botanical Garden. It was once an itty-bitty seedling, so how did it get this big? The answer is secondary growth. And that's the process responsible for the lumber in our homes and for trees that live for millennia. Today, we'll be peeling back the bark to understand how plants bulk up through secondary growth. I'm Dr. Joyce Onyenedum, and I'm a botany professor at New York University. Welcome to Build a Plant, my video series where we discuss the fascinating hot topic of plant anatomy. Secondary growth refers to the lateral expansion of plants and occurs in everything from tomato plants to tree trunks.
It evolved at least 3 times independently in lycophytes, ferns, and seed plants. Secondary growth was subsequently lost in ferns and therefore is not present in any extant species. And within flowering plants, it was lost in multiple lineages, most notably in the monocots, the group that includes grasses and palms.
As a reminder, in the previous episode, we discussed primary growth, which adds length and new organs to plant shoots, thanks to the activity of an apical meristem at the tip of the shoot. Secondary growth commences lower down on the shoot axis once primary growth has finished adding new organs. It occurs through the activity of 2 lateral meristems, the vascular cambium, which generates vascular tissues, and the Phellogen, which produces the outer bark. First, let's talk about secondary growth via the vascular cambium. This developmental process provides mechanical support to plants as they grow taller and an efficient means of water and nutrient transport. The vascular cambium is a bifacial lateral meristem that produces the secondary vascular system, which includes the secondary xylem, aka wood, and secondary phloem, aka inner bark. The activity of the vascular cambium allows plants to grow in girth. It originates from 2 sources, the fascicular cambium from procambium within vascular bundles, and the interfascicular cambium from parenchyma between bundles. Because the vascular cambium arises from a ring of primary vascular bundles, some botanists actually speculate that the lack of secondary growth in monocots is due to the fact that vascular bundles are scattered throughout the stem. Let's take a closer look at secondary tissues produced by the vascular cambium. Secondary xylem, commonly known as wood, is primarily responsible for water transport and structural support. It typically comprises conducting cells, such as vessels and tracheids, and also supporting cells like fibers, and both axial and radial parenchyma.
The arrangement and proportion of these elements can vary across taxa, influencing wood properties and function. Secondary phloem is composed of sieve elements and companion cells for photosynthate and signaling molecule transport. Parenchyma cells, both axial and radial, and sclerenchyma in the form of fibers, sclerids, or both.
The phloem structure is tied to its function and long-distance transport and plant defense. Next, let's talk about secondary growth via the Phellogen, a meristematic region that produces phellum outwardly and phelloderm inwardly. The Phellogen, phellum, and phelloderm are collectively called the periderm, the outermost protective layer that replaces the epidermis in stems and roots undergoing secondary growth. Many researchers refer to the periderm as the outer bark, and the secondary phloem produced by the vascular cambium as the inner bark. The outward appearance of the periderm is determined by numerous anatomical characteristics, such as the layers of phellum, as well as the amount of parenchyma or fibers present. Take, for instance, the green, vivid bark of striped maple. It's photosynthetic. The cells of the periderm are full of chloroplast. The periderm provides protection against water loss, fires, pathogens, and mechanical damage. These adaptive features are crucial for plant survival and have been exploited in various industries. For example, wine corks are made from the periderm of Quercus subra trees. Now let's explore the exciting application of secondary growth, dendrochronology, the study of tree rings. This field merges the realms of plant biology and climate science. Dendrochronology relies on the fact that the vascular cambium responds to both genetic and environmental factors. In temperate trees, early season secondary growth typically produces cells with larger diameters and thinner walls, while late season growth results in smaller cells with thicker walls. This environmental response is seen in the anatomy of secondary xylem, forming distinct annual rings. The width of growth rings of living species, and even in fossilized trees, are used by researchers to reconstruct historical climate conditions. An unusually thin growth ring, for instance, could correspond to a particularly dry year or even an infection. This application of wood anatomy showcases how the study of secondary growth extends far beyond plant biology, offering insights into broader ecological and environmental changes over time. But when you thought you had secondary growth all figured out, nature throws you a curveball with cambial variants, fascinating modifications to the typical secondary growth pattern. Cambial variants arise from abnormal development, distribution, and abundance of secondary vascular tissues. Intraxillary phloem is one example. In the stems of Campsis radicans, trumpet creeper, we find secondary phloem being produced by the vascular cambium outside of the secondary xylem as per usual.
However, there is also more secondary phloem made at the juncture of the primary xylem and pith.
Scientists think this excess secondary phloem helps climbing plants achieve flexibility as they twist skyward. Cambial variants in general are a common feature among independently evolved woody climbers. Successive cambia are another example. They superficially resemble growth rings, but are something entirely different. There are actually multiple vascular cambia that form concentrically encircling the stem. These additional vascular cambia arise from parenchyma-rich regions that have the potential to de-differentiate and become meristematic again. As with intraxillary phloem, this change to ordinary secondary growth helps climbers to bend their woody shoots. Let's recap. Secondary growth is the process by which plants expand laterally and develop the thick trunks and branches that support their impressive form. It is achieved through the activity of two bifacial lateral meristems. The vascular cambium, which produces secondary vascular tissue that enable the plants to grow in girth while efficiently transporting water and nutrients. And the Phellogen, which produces the periderm, the outermost protective layer against environmental stresses and damage. Secondary tissue anatomy is intimately tied to plant function and adaptations to environmental variation. It can get pretty extreme in cambial variants and form the basis of entire fields of science like dendrochronology. In our next episode, we'll shift our focus to leaves, their incredible anatomical diversity, unique adaptations, and essential role in photosynthesis and plant survival.